Hello, everyone. Thank you for joining our webinar this month. I am delighted and honored to be able to do this webinar for you guys today and really excited. I just want to share a little bit about the passion that I have for our company, Microbial Insights, the history of the company, how we got started, and then to kind of share with you a little bit about where we plan to go and what we're going to be doing in the future, and also introduce you to the amazing affiliates that we work with around the world. I know it's a trying time, so I appreciate that everyone's taking time out of their schedules to be here with us today. So almost 30 years ago, the idea to create a commercial laboratory to offer the latest molecular tools to really look into the black box of remediation and assess and quantify different microbial populations resulted in the founding of Microbial Insights. We were a technology transfer company from the University of Tennessee based on the world-renowned work of Dr. David White. Many of you around the world know Dr. White. He was an amazing researcher. He was both a medical doctor and a PhD, and his vision was to take the molecular tools that were developed and validated in the medical industry and transition them to environmental remediation. In the medical industry, they have money to be able to develop tools, but as you all know, in remediation, that's something that we really don't have. All right, sorry about that, having a slide issue here. So let me get back into Dr. White. You can see his picture here on the screen a little more. Dr. White was very passionate about our natural world and having a positive impact on the quality of the environment. You know, his inspiration really created this place, and to be honest, gave me and many other well-known scientists the motivation to pursue careers in the field. So it's my goal as one of the owners at Microbial Insight and the president to continue his legacy, and I really want to have a positive impact on our world. So we're not based in Knoxville, Tennessee as a gimmick or to ride the coattails of someone else's reputation. We're based here in Knoxville, Tennessee because 30 years ago, this is where it all started for our laboratory. So I don't want to get your samples because someone Googled wrong and ended up shipping to our lab, or because the sampler mixed up the address and used a Knoxville location. I want to get your samples because we earn them. So here's what we do. I want to go into that a little bit for you. Not only in the U.S., but around the world, we really want to earn your business. Quality matters to us. You know, we want to consistently produce accurate and precise data that meets the highest QA/QC standards, so we'll talk about that today. We also know that expertise matters. I want to give you 100% unbiased data and interpretation to help keep you informed about your management decisions. And then I want to show you how people matter to us. You know, as customers, you're very important to us. We want to give you the best customer service, and we want to work with the highest affiliates that we can around the world. So we are truly unbiased data. Microbial Insights is not owned by or directly affiliated with a consulting firm. We work with every consulting firm that's there. No one firm is more than 7% of our business volume. So whether it's the very large firms that own agencies all over the world, or it's the mom and pop locations that are working in small locations, we work with everyone out there equally, and we're not owned by anyone, so your data is controlled by us and is not something that's subject to being disseminated to other firms. We also don't sell any cultures, and we don't sell any products or amendments. We give you the very best data along with analysis interpretation, but we're not trying to sell you cultures or microorganisms. Based on the data from your site, we want to help you come up with a remediation solution, but we don't have any conflict of interest. We work with every vendor with their products, and our only incentive is to help you, the client, make the best possible decision on the best possible data, and we are a certified woman-owned small business. So we do have the most experienced and qualified staff of any lab. I'm really proud of the work that we've done. And as I mentioned, Microbial Insights was founded in Knoxville about almost 30 years ago. And I've been with the company for 20 years. My role as president is to continue Dr. White's legacy, to continue the great things that he's doing. I've seen a lot of evolution of molecular tools and a lot of site data. And I'm so proud of the work that we do in our industry. And then many of you, I'm sure, have met Dr. Kate Clark, who is our technical director here at Microbial Insights. Kate is an amazing person. She has a very sweet personality, and she's very knowledgeable on every molecular tool that's out there. You'll find that Kate's very good at advising you on the different solutions that are available for your sites and your projects. So if you have questions, Kate's someone great to reach out to. And then we have Dr. Sam Rosalina, who is the director of our Environmental Forensics Division. Sam is an amazing young scientist and a brilliant person. When it comes to chemistry, there's no one out there. When it comes to compound-specific isotope analysis, he knows that tool and how to get good QA/QC, so it's quality data that you can trust on these analysis so that you feel good about the data that you're receiving. And then we have Casey Brown, who is the director of our Omics division. Casey is also an amazing scientist, and she's very hands-on. You'll find her in the lab, whether we're talking about genomics, metagenomics, transcriptomics, proteomics, whatever it is. Casey is in there hands-on working with the technicians to make sure, again, that you get good, high-quality data. And then last but not least, we have Jennifer Amos, who's our Director of Customer Service. And Jennifer is like no one else at being able to organize and make sure all of your sampling supplies get to the site. And she's always thinking about you guys as customers and what you need in the field. So hopefully you always feel like you get the best customer service from us, because I know that's Jennifer's goal. So we are fastidious scientists that are wearing our lab coats, working in the lab, but we're also friendly and accessible people. We're proud to provide solutions for you guys, our amazing clients, and we're proud of the fantastic feedback we receive each year. You've probably seen us send out a survey to our clients, and based on the results of hundreds of clients, they had 100% satisfaction. We really didn't have any complaints in our surveys. And the words that you guys said really made us feel good, innovative, reliable, service-oriented, and that's what my staff and I want to be for you. We also want to be sure that you get the highest level of QA/QC in the industry and that the results are accurate. So for 3 decades now, the primary mission at Microbial Insights has been to provide the most accurate and precise data in the industry so we can ensure that our clients can use our results as an integral part of site management decisions. So as part of our commitment to data quality, Microbial Insights participated in a federally funded qPCR study a few years ago. And what happened in this study was a known concentration of the Halococoides. You can see it on our chart here. This is an example of the high concentration. So we had 5 rounds with 5 high concentration replicates, 5 low concentration replicates, and 2 blanks. And each round you can see as an example from one of the high concentration rounds, this is what the results looked like. Involved in the study were 2 academic laboratories, a national laboratory, a North American competitor, and then Microbial Insights. And you can see the 5 green data points from MI are so close that it's difficult to even tell the results. And these are separate qPCR extractions, so extracted DNA from individual samples all the way through the process. Every time, all 5 rounds, all 10 samples, both high and low concentration, our replicates were on top of each other like you're seeing here. So some of the labs, as you could see, had a lot of variation. They were losing samples. Some of them had contamination in their blanks. Some of them could not tell a difference between low concentration sample and a blank because they were losing it in the extraction. So being sure that you have high quality assurance is really important to Microbial Insights and to our industry so that we can trust this data that we get. So I would really encourage you to make sure that you check your lab, check the QA/QC, and make sure you trust the data that you're getting. Furthermore, to ensure that you get accurate results, all our DNA and RNA extractions are performed on samples the day they're received. Even if you send them to us on Saturdays, we have staff here that are ready to extract your samples. And then to ensure high quality assurance and the integrity of our results, we always perform extraction blanks so that we can test and make sure that there's no cross-contamination. We run negative controls on every plate. And as with an extraction blank, detection of a target gene in a negative control is direct evidence of contamination and would invalidate the sample results. A positive control is also included on each and every plate so that we can confirm PCR amplification and can act as a continuing calibration check. So I want to be sure that the data that you get is consistent round to round, even if you're talking years apart in the sample analysis. So these measures obviously cost money, And to be honest, I found that they're neglected not just by the startup laboratories, but by labs you would expect to have better QA/QC. But we feel like you guys are worth it, and I personally feel like my reputation is worth it, so I would hang my reputation on the results that we produce. And then if you've heard me talk about the database. I am so proud of the databases that we have available. You know, with over 30 years in the industry, we've seen more samples, any sample matrix that you can imagine. So our database contains more than 50,000 samples from all 50 states, all U.S. territories, all 7 continents, and more than 55 countries around the world. And as our client, you have exclusive access to this valuable resource. So in the qPCR database, you're going to find over 150 unique organisms and gene targets from all these different locations, so you can start to put some context to your data. What does 10 to the 3 mean for this particular organism or for this functional gene? Is that a high concentration or a low concentration compared to sites around the world? Then in our CSIA database, you can automatically plot data with a lot of customization to be able to have great visuals that you can use for interpretation. And also Sam's group does a great job of making sure to update those literature values. So whether you're talking about enrichment factors or isotopic factors, there's over 500 literature values that are in our database that are continually updated. So you've got one location that you can go to get all that information rather than having to dig through papers on your own. And then we're constantly innovating and expanding. I'm so excited about the new opportunities that we've had. You've probably heard about our recent acquisition of Pace Energy Services, where we truly are expanding and growing. We've expanded our CSIA capabilities, so Sam's area has really grown and what we can do in there, along with petroleum forensics. If you have questions on that, be sure to reach out to Sam and talk to him. And then we're not only leading the industry with state-of-the-art equipment, we're advancing the field of diagnostics. The foundation of our innovation is our engagement with research and development. So we work with researchers around the world to try to commercialize new technology that they developed, from work on metabolomics and proteomics to development of new tools to evaluate mineral transformation. We're changing the field to increase the speed and effectiveness of the remediation strategies that are available to you guys. So you don't have to wait months to get the research from a university. We try to commercialize that so it's readily available to you. And we're continually involved in grants on our own to develop new tools and make them accessible and cost-effective for use in remediation. We've been funded by NSF, NIH, CERDUP, ESTCP, just to name a few. And then finally, we're a trusted partner. So when you work with us, you get more than data. We want to be part of your team. We want to give you peace of mind. We're a trusted partner for government. We work with all the government agencies. We work with the end users, different industries, whether it's paper and pulp, oil and gas, chemical manufacturers, pretty much anyone that has remediation needs, we are involved with them. We also work with academia and the military very closely. So since 1992, we're proud to be able to provide our amazing clients with solutions that support a sustainable environment and strengthen the communities that depend on it. And especially during this difficult time, we find that that's very important for us to be able to serve you. You know that we're a leading provider of tools for environmental market, but our reach is a little further. Some of the industries we commonly work in are microbiologically influenced corrosion, so we're trying to protect assets in the oil and gas industry and the paper and pulp industry. We do work with microbial source tracking, where we can determine the host microorganism that is contaminating the surface water. We do work with agriculture, looking at runoff, different crop yields. We do work in landfills, biofuels, different wastewater, and then art restoration and astrobiology, which I think are really interesting, and I want to share with you just a little bit on those projects that we've done. So many of you probably don't think about the microbial needs that come with art restoration, but we've been very blessed to work on some really, really, truly interesting projects that can have, impact at World Heritage and cultural sites. So we've done work on the Silk Road in China, on the grottos, looking at fungal formation that was destroying these artifacts. We've done work on the buildings at the Salk Institute and the Ames House. And then we did a really, really interesting study with King Tut's tomb that sometime reach out to me. I'll be glad to share with you the story on that. But trying to understand the fungi that's growing on the walls in the tomb. Was that ancient fungi that came when that tomb was originally sealed, or did that come when researchers entered that facility later? And then over the last 30 years, we have worked a lot with NASA looking at astrobiology, and Dr. White was very intimately involved with a lot of the researchers at NASA and trying to help develop tools that they could use. And I've been blessed to be able to educate some of the scientists there a few times and be involved with them on projects, whether we're trying to replicate environments similar to other galaxies, like looking at desert and extreme environments like the Mojave Desert, the Death Valley, or volcanic activity. We've also been involved with them looking at the other extreme. What if it's a super cold environment, looking at Antarctica and the dry valleys, or Arctic permafrost, and then they can translate that to what we might see on other planets. We've also been blessed to get moon rocks, been able to hold those in my hand and to be able to do microbial characterization of those to understand what's there. And then finally, we've done work with them on some of their chambers, like the Mars chamber, looking at gas leaks that were causing microbial growth within these systems. So really exciting the work that we can do and how important microorganisms are in every process that you can imagine. If you think back to the Deepwater Horizon spill, the Microbial Insights biotraps were deployed by robots, as you can see in this picture, 5,000 feet below the ocean surface. We've also had divers that have taken our biotraps and piezometers and deployed them in the ocean floor in the China Sea. So they actually dove down and drove those into the ocean sediments in the sea there. And why do we do all this? Well, I was blessed a few months ago to do a talk with the National Institute of Health, thinking about how our human gut microbiome has changed. So when we look at agricultural runoffs, the changes that we're making in soil, the industry changes that are happening in our world, we're affecting surface water. Urbanization is definitely changing our planet. And then animal health, we're adding chemicals and antibiotics, growth hormones to our animals. And all of those ultimately impact our groundwater, which is why we're all here trying to make that clean, because ultimately that impacts our human health. And we've seen dramatic changes in our human gut microbiology over the last couple of decades. So these tools are great to be able to help us understand that and take better care of these natural resources. You know, groundwater is one of the most important natural resources that we have, so we all want to protect it. So as I mentioned, we have locations all over the world. Again, we're headquartered in Knoxville, Tennessee, not because Knoxville is the shipping hub of the world or because Tennessee has the strictest environmental regulations, but we're headquartered in Knoxville because that's where Dr. White's work originally started and where our company was founded. So we want to honor that, and we keep this headquarter location to honor his legacy here. But we also receive samples, as I mentioned, from all 50 states, all U.S. territories, 45 different countries, as you can see on this map here. And then we have locations, and I'm so excited to introduce you to the leaders that run our affiliate locations around the world. So now I've kind of shown you MI as a whole. You can see the map here. We're going to talk to reps in our location in Canada, Germany, Belgium, Italy, and then we also have reps in China and Australia that I will tell you about in just a moment. So first up, I want to introduce you to the rep from our other North American location. We're going to have first up is Will Lush, and I've worked with Will for almost 20 years. You know, Will's dad was Don Lush, and he was one of the original investors in the creation of Microbial Insights. He was truly an amazing man, and he and Dr. White were so fun to be around because they were such inspirations for our industry. They really cared and were passionate about bringing these tools to the remediation market. And Don Lush was personally a trusted advisor and mentor for me for over 20 years, and I miss him terribly. So I'm excited to have Will here with us today. Will owns a company called EBPI that's located in Toronto, Canada, and his lab is the receiving point for samples that come from Canada. EBPI was established the same year as Microbial Insights in 1992, and they are a world leader in toxicity testing. So I'm going to turn it over to you, Will, and let you tell us a little bit more about what you're doing at EBPI and what's happening in Canada.
Thank you, Dora. I appreciate that. I think you took most of my points from the first slide there, but we'll run with it. I'm happy to present a few slides on EBPI, or environmental biodetection products, today. I was originally thought about presenting maybe some of the projects that EBPI and MI have worked on closely together over the years. Though I think, for this presentation, I think sort of a high-level overview of EBPI's technologies and products and services may be the best fit. So who is EBPI? Well, EBPI is a small biotechnology company based out of Burlington, Ontario, and we are the representatives of Microbial Insights for the Canadian market. And as Dora said, around the same time, I think it was 1992, that Microbial Insights was forming, EBPI was also beginning to take shape under an environmental consulting company called Beak International, and under the guidance, again, of Don Lush, who was also a board member at Microbial Insights. So, like Microbial Insights, the concept of EBPI is based around adapting molecular tools and techniques, but we focus on monitoring for toxicity and subchronic toxicity in the environment. So currently today, EBPI develops, manufactures, and distributes testing products to over 40 countries. And we currently have over 15 international distributors that are more dedicated, and we do have work with other distributors as well in different areas. We have a fully operational lab to perform client analysis in-house under GLP guidelines, and we're currently working towards ISO certification if that's required for your projects. So today our focus hasn't changed since the inception of EBPI, as we continue to focus on the research and development, bringing new tools to measure the health of the environment. So what I'm going to focus on again is 3 areas of EBPI's platform. So we have a lot of other tools at our disposal. I'm going to focus sort of on toxicity testing, genotoxicity testing, and mutagenicity testing at a high-level overview. Slide 2 be great, Dora. So when we talk about toxicity testing, we talk about, is an organism dead or alive? Can we calculate an LC50 and LD50? So what we do is we offer 2 different bacterial test kits. First being EBPI's E. coli test kit. So on the picture on the bottom left there, we're looking at a rough strain of a rough mutation strain of an E. coli strain, a laboratory strain, which naturally produces a high level of beta-galactosidase. And we expose this bacteria to a potential toxin. And after an initial incubation period, we add a chromogenic substrate. And if the bacteria is alive, we produce a deep blue color. And if the bacteria is dead, it's a very clear color, no color develops. And from there, we can read this quantitatively or qualitatively. In the last couple of years, we've worked with a group out of Finland to develop various Vibrio fischeri toxicity tests, which anybody in the area of toxicity tests are probably familiar with the Vibrio fischeri. They're typically usually ISO standards and regulated for discharge of fresh water. So in the last couple of years, we've helped develop a 96-well format for the Vibrio fischeri test, which is now an ISO standard, which is the ISO 21338-2010. And this allows for samples to be screened, matrix samples that may have high turbidity quickly and easily. We also offer replacement bacteria or in-house testing for the Microtox M500 line. We've also developed test swabs that can be taken out of the field on site for measuring toxicity around gas and oil with the Vibrio fischeri for quick measurements and quick results. And we also have sediment tests for both our E. coli and Vibrio fischeri tests if required. You can continue, Dora. Slide 3. So what I really want to focus is our expertise comes into not just toxicity, but subchronic toxicity or genetic toxicity. So when I describe genetic toxicity or genotoxicity, we're looking at a sub-level of toxicity. Here we're not measuring is the bacteria or is the organism dead or alive. We're measuring the amount of DNA damage. So the two tests shown here are two different genotoxicity tests that EBPI offers. And both of them are measured or designed to measure how hard is the bacteria or cell line attempting to fix damaged DNA. And the one in the blue is a SOS response, and the one in the yellow is a UMU response. They both are different bacterial strains. So if you're looking for a genetic toxicity test to measure oxidative damage or something else at a site or water treatment facility, these are great screening tools. As it's noted here, you can see that one is an ISO for water determination of genotoxicity in water and wastewater. And we've started to see some regulation internationally with them. Germany is regulating wastewater effluent with the UMU test. And we're starting to see some non-regulation adaptation in France and Germany, Sweden, Holland, and throughout Europe, which is good to see. Next slide, Dora. So we've gone from toxicity down to genotoxicity. Now we're going to mutagenicity, which is a subset of genotoxicity. We're going further and farther in. So here we're not measuring how hard is the system trying to repair damaged DNA. We're actually measuring frame shift mutations or base pair mutations within the DNA. So we've taken a 1980s approach, which is an Ames test, which is highly published at this point, which is on the bottom left photo. We've adapted that and transformed them into 3D4 well or 96 well plates, which are much more sensitive. What we're finding right now is that the 3D4 well plate is an ISO standard, but we have a lot of clients right now in the drinking water and wastewater industry that are using our 96 well plate. And they're coming back to us and saying it's much more sensitive than the 3D4 well plate. And so we're quite excited about that we've developed a more sensitive assay than any of the other mutation assays out there. The other one I wanted to talk about was our Ames Express strains. A few years back, we worked with the University of Guelph to incorporate human recombinant enzymes into our bacteria strains. So we wanted to see what type of compounds are metabolized through different metabolic pathways and look at the sensitivity. Currently, most assays use either a rat liver or a hamster liver for mutation assays. But if you look at halogenated compounds, when they're metabolized through our GST theta 1s, they become highly reactive species, which we just don't pick up the detection with a rat liver or a hamster liver extract. So these strains are highly sensitive for whichever whichever compound you're looking for. P450-182, which is highly sensitive to various pHs. And again, our GST theta 1, which is very, very sensitive to halogenated compounds in water samples. Slide 5, Dora. So who is EBPI? Well, where have our kits been used? The scope of EBPI, we've been lucky enough to work with the EPA during the Gulf of Mexico oil spill. So our products have been down to the bottom of the oceans, and we've placed our product up onto the International Space Station to measure the rates of mutation and how they differ in outer space than on Earth. And our products have been used globally in over 45 countries on Earth. It's an interesting note here. During the Gulf of Mexico oil spill, the EPA mandated the rotifer test, which we had been looking after out of Belgium, to be used whenever the dispersants were being used at the Gulf during the Gulf of Mexico oil spill. There was a lot of data looking at the toxicity of the dispersants. There was a lot of toxicity data associated with the oil, but nobody had, my understanding is they're looking at the mixture effects of the two. So whenever the dispersants were being used, the rotifer toxicity test was being used to measure the toxicity associated with the samples. And at the same time, we were working with various universities around the Gulf of Mexico. And in one particular was the University of South Florida. And we were looking at mutagenic byproducts that were being produced from the same waters that they were extracting from the oil head. And what came back was that we started to see high rates of mutagicity associated with the water surrounding the oil head, but we saw low levels of toxicity. So what we really want to do and focus on is taking our research and saying, well, we can stop looking at toxicity, and we really want to start looking at subchronic toxicity, low levels, in an easy format. So our main area of focus, and though we do have a lot of clients, we test for new compounds and new pharmaceuticals, we do focus on air particulate testing. Our kits have been used in various EPA, USGS, Environment Canada projects for measuring the presence of genetic damaging compounds in air particulate. We've also worked on FDA projects looking at various effects of cigarettes versus vapor products versus e-cigarettes. In the area of drinking water and wastewater, though most of our toxicity tests are used in the wastewater industry, our genetic toxicity tests are being quite adapted nicely in the area of drinking water. We're working with universities globally, private corporations from Canada to the United States, South Africa, and throughout Europe and Asia for both monitoring the genotoxic effects of drinking water. So why is this important? Well, what we're seeing here is with the growing population, and especially in the US, we're starting to see the concept of toilet to tap coming to reality. And this is due to limited fresh water resources and an increasing population. So we have these new chemicals being released into our environment, making their way back into our source waters, and this is becoming a huge concern. So to ensure a healthy population, research into drinking water technologies and various products and byproducts is becoming an increasing concern. So our products are sort of an ease to use, low cost analysis to fill this void. So we have a lot of analytical tools and to measure various compounds in the environment that we know what we're looking for, but we don't have a good screening tool, or we're not using the great screening tools for measuring the mixture effects or the non-regulated compounds that we don't know about yet. So some of the tools and services that we offer are regulated in various industries, from the environmental remediation industry. These tools are assisting to sort of make a strong educated determination for your sites. So we can look at the mixture effects of things and say we're not concerned about it, even if some other compound may be there. The metabolites are being metabolized, or the compounds being metabolized, and helping look after your site. So if you have any questions regarding EBPI's lines of kits or services, please don't hesitate to reach out to Dora or contact myself at sales@biotoxicity.com. Thank you, Dora.
All right, thank you so much, Will, for that great presentation. Next, we're going to move on to Europe. First up is Dr. Stefan Hoopman, who is our partner in Germany. Stefan is truly a brilliant scientist. I think you're going to really enjoy his talk today and what his company has to offer. He and his staff run our molecular division in Germany. The company is renowned in Germany and comprises process technicians, engineers, biotechnologists, hydrogeologists, soil scientists, geologists. So they're truly an interdisciplinary team of individuals that cover and do work with our company in Germany and in Switzerland. So I will turn it over to you, Stefan.
Thanks, Dora. Yes, hello, everyone. Yeah, I'm really happy to introduce Sensatec as a company based in Germany with branches in different parts of the country, and also partners in different parts of the world. We're mainly active in Central Europe, though, also tending some work in Africa. As basically the core markets. Currently, we are working on approximately 150 projects. Next slide, please, Dora. Yes, and if you hit it one more time, right? Thank you. Yeah, our core business is in applying in situ remediation technologies. We started out with in situ biotreatment, aerobic, and later on a lot of anaerobic treatment. A strong focus for the last 10 years, I guess, in Central Europe has been on chlorinated solvent sites. So that got us into a strong involvement with reductive dechlorination. But nowadays, a strong trend is towards ISCO technologies, chemical remediation using phentons, permanganate, persulfates. It's very popular nowadays. So this is what we do, and eventually also physical remediation technologies like mobilization technologies and also thermal treatment. Yeah, next slide is great. So there seems to be an automatic continuation of the slides. One aspect of doing soil remediation in Central Europe is about there's no more easy sites left now. We are looking back at say 25 years of soil remediation, so all the easy sites that had been have already been remediated. So basically, you will not find any more substantial contaminants in sandy structures that are easy to remediate even with hydraulical means. So we're now focusing strongly on clay and silt structures, on fractured bedrock and heavily layered aquifers. So we need to find solutions for that, which brought us into the field of solids emplacement using high pressure injections, injecting, for example, sand structures, as you see on the bottom picture here, into clay formations, using hydrofracking, or using pulsed injections, basically to get access to contaminants and to be able to remediate even very demanding and difficult sites with difficult geology. Next slide, please. This eventually also got us involved in direct image drilling technology using geoprobe technology. So we are operating a couple of differently sized geoprobe rigs from small to large scale. That will enable us to identify contaminant locations and a quantification of contaminants in much more detail. We think this is also a really helpful tool to identify relevant sampling locations, also for molecular biology tools. So we think the trend towards a more detailed investigations in the lab is a wonderful tool and can be accompanied by a very detailed site investigation tools such as direct image drilling technology. Most of these tools are focusing on organic contaminants, but we also have a very strong partner in Germany that provides us with an XRF tool, which is basically a X-ray based fluorescence tool that we can use to detect heavy metal and metalloid concentrations in soil structures. Apart from that, of course, there's the other sensor-based tools such as electrical conductivity, cone penetration test, to identify soil structures as we drill through a formation, or hydraulic profiling tool to get a better understanding of the hydraulic properties. And we are really happy to have teamed up with Microbial Insights, because I think the combined use of detailed lab investigation tools together with direct image drilling technology will really, for us, means a major step ahead in identification of relevant processes on the ground. Next slide, please. Yeah, last but not least, we also offer the production and delivery of microbial cultures for soil decontamination. We're happy. A few years ago, we're lucky to acquire a company with more than 20 years of experience in this field. So we constantly keep up cultivations of different types of microbes. Aerobic and also anaerobic ones like Dehalococcoides culture that will enable us to provide different types of microbial cultures with specific degrader capacities 24/7. Last slide, please. Yeah, basically the set of range of services enables us to provide our customers, which is basically major industrial partners in Central Europe, with technical services that eventually are meant to provide remediation success by not looking at the site in a superficial way, but providing very sophisticated technologies and services to fulfill the customer needs. And with that, I would like to end, and I'll be available for questions later if there are any. Thank you.
All right, thank you very much, Stefan. So next up, we're going to continue in Europe and talk to our sales rep in Italy, Claudio Sandrone. Claudio is our sales rep there, and if you've met Claudio, you really can't help but love him. He has such an infectious personality, so I'm delighted to have him speaking with us today. Claudio owns a company called BAW, and it's an environmental design and consulting company that focuses on integrating the very best in remediation technologies that are available in the market, and he uses that to create the most advanced solutions for environmental problems there. So I will turn it over to you, Claudio.
Thank you, Dora. Hope you can hear me well now. Good day,
yes,
everybody.
I can hear you.
Okay, I'm glad to give my presentation in this International Microbial Insights webinar today. I'm the Business Development Executive of BAW, and I'm an environmental engineer. I've been working in the remediation field since 1997. That makes 23 years so far. I was the founder of TRS in 2001, an Italian company that became in few years one of the greatest remediation companies in Italy, with more than 100 employees and a turnover of about 15 million euros. I sold the company in the middle of 2010 and left it in 2014. In 2012, I founded BAW, and we are an environmental engineering company, as Dora told, based in Saluzzo, near Turin. Here you can see our services. Next slide, Dora, please. We do remediation design, environmental characterization, GIS, due diligences, environmental consulting, but we are very focused on innovative technologies. I have spent a large part of my professional life looking for new and advanced technologies around the world, particularly in the United States and Canada, doing meetings with universities, research centers, and manufacturers to identify the best products that could be launched on the Italian and European markets through meetings, conferences, projects, and so on. In 2012, BAW becomes the entity to house the product line, becomes the core business of the company, alongside traditional engineering activity. Next slide. The partnerships developed with international companies, guaranteed by representative agreements in Italy and Europe, are the real strength and the entrepreneurial spirit of BAW. This spirit is well summarized in the TTE, Technology That Excite program, that is now the flagship of BAW, an international project aimed at promoting the best remediation technologies currently available on the world market. The project brings together leading-edge technology companies within an international network that is offered to industry from which they can find the best technical solutions for their environmental issues. A sort of technological hub, as we like to say. You can see more about TTE program on our website. And thanks to technology, the TechSite program in 2017, BAU was awarded in the Italian government palace of Montecitorio in Rome with the prestigious 100 Eccellenze Italiane Award as one of the best 100 companies in Italy. Starting from the beginning of 2015, we became representative in Italy and for some of them in Europe of these 6 following companies. IVE International for surfactant and mediation technologies, that is a Canadian company. Then Provectus Environmental Products, you know it is an American company that deals with innovative technologies for remediation of contaminated soils and groundwater. Geolog, that is a German company that offers advanced systems and tools for environmental characterization and geotechnologies. Then we represent Sensotech. You have just listened to Stefan with innovative remediation technologies and bioremediation. Then we have QAD with passive sensors for groundwater. And then the last but not least, Microbial Insights with biological analysis and tools we represent them for Italy. Why we choose Microbial Insights to be our strategic partner in TTE program for microbial analysis and tools, I will show you the next 3 slides and we will understand why. In Europe, we have 3.5 million of potentially contaminated sites. Of these, more than 1 million, they have been confirmed as contaminated, and less than 10% were remediated. We know that soil and groundwater are non-renewable resources, and for this reason, they must be preserved. We need a collective effort that action needs to be undertaken at all levels, and new technologies must be pushed through investment in research and knowledge. And if we are here today, it is because I think each of us is already engaged in this regard. Next slide. Where is the market more active and dynamic? In Italy, we have the highest overall turnover, about 1.6 billion euro per year. We also observe high turnovers in all the countries with the highest degree of regulation: UK, France, Germany, and Netherlands. The blue area indicates the nations where there are large companies operating at an international level. Italy, on the contrary, suffers from dwarfism from this point of view. High demand from the private sector and a high variety of applied remediation technologies. We are here in the market that I would define mature market. Instead, in the gray area, we have states with fewer companies in the market and lower turnover, where the market is still driven by government intervention. Today, the vast majority of interventions in Europe in terms of value is the physical chemical. 33% is still big and dumb, and only 90% is given by biological treatments. But in recent years, we are seeing a rapid growth, perhaps due to the global crisis and hence the use of ability of funds of the in situ treatments, much to the advantage of biological ones. The future is going in this direction, and I think it's very positive. Now you know why we chose Microbial Insights. Let me say, I love Microbial Insights. It was a love at first sight. I would say I found Microbial Insights the perfect partner for microbial analysis. With their comprehensive reports, with their real-time answers to our emails, with their professionals, and with their huge database. They have over 50,000 samples from sites around the world, so they can really understand and put context to the data. The Microbial Insight database, if you are a customer, can be a really good research to help you understand what that data that you get means. Well, coming to the end, you can find my contacts. We believe, and that has become our motto, this wise saying of Native Americans, that we borrow this earth from our children, and since it is so, each of us, we are here for that, must do his part. Thank you, Dora, and thank you all for your attention.
Thank you, Claudio. Hearing you speak makes me want to go back to Italy right now. So we appreciate you doing it. And if you have questions for Claudio, go ahead and enter them into the software, and we will get back with Claudio at the end. Okay, so finally, last but not least, I want to introduce Luke Gart, who is with our, sorry, hold on just one moment. Let me make sure Luke Gart can be heard. Okay, we have Luke Gart, who is with our affiliate in Belgium. For the past 10 years, our lab partner in Belgium is Avicom. Avicom was founded in 1995, and they've developed a wide range of products for wastewater and aquaculture, as well as bioremediation. So they have a wide range of cultures that are available, and they perform many lab and pilot scale feasibility studies. They're one of the most respected laboratories that you'll find in Belgium and the Netherlands. They have an amazing reputation there. So I'm really honored to have Luke Gart here, who's the Senior Project Engineer. So I'll let you go ahead and get started, Luke Gart.
Thank you, Dora. Thank you for this amazing introduction. This is a very interesting webinar. I nearly forgot I'm a speaker myself. But now I'm happy to introduce Avicom with a few slides to present what we're doing. So as it is written there, you can see that Avicom is an innovative R&D driven small company, and we are specialized in steering and optimizing microbial processes. And I would like to stress the word microbial processes because we try to involve microbes everywhere. Every time we have a new project or something, we start with bacteria and we see what bacteria can mean for us, for nature, for people, where they can be of added value. So we always start from bacteria. And we use the bacteria in different kinds of applications for soil remediation, but in projects with water treatment, but also the production of single cell protein to replace the meat, all the meat that we are using. We also have a culture that is used in aquaculture. So we have quite a variety of application areas, but it all is related with microbial processes. Next slide, please. So you can see there a picture of Willy Verstraete, who founded Avicom. And he is, some of you will most probably know Willy Verstraete because he's a world top microbiologist. He was a professor at the Ghent University, and he was always, as a professor, looking to find new applications with bacteria to create added value. He's always looking for added value. Where can bacteria help us? Because bacteria have a lot of potential. They can mean so much more than we think. So he's always looking for added value. You can see there also a picture of our analytical lab, but we have, we're a small company, but we have an analytical lab, but we have also facilities for the microbiological lab, a level 2 lab. We also have upscaling facilities. We have a pilot hall for testings for processes on cubic meter scale. So we have quite some facilities here. Yeah, next slide, please. So what are we doing? What is our core business? In fact, we have 3 kinds of businesses. We do projects for industrial partners. And this is really tailor-made. If we meet industrial partners with a certain problem and we think we can be of added value, we go and talk to this partner and we try to find a solution for a particular problem. So we work for industrial partners, but we also do innovative research via subsidized projects. I'll come back to that later. And a third point of our core business is that we develop and we sell bioproducts applications for example, our aquaculture, but also single cell protein applications and so on. Yeah, next slide, please. This is a picture of our team. We are 7 engineers. 3 of them have a PhD, and I'm the person there, the lady in the middle with the pink pullover. But this is our team. Yeah, next slide, please. So I cannot introduce all the topics that we are working on, but I thought to highlight a few of our topics, of our cultures. And one of the cultures that we produce, we developed and we commercialized the multidichlorobac culture, MDB, which contains dihalococoides. It's a culture to degrade chlorinated ethenes, PCE, DCE, vinyl chloride, but also 1,2-DCA. And this culture was in fact developed starting from a contaminated site where there was a lot of 1,2-DCA. So we started to develop that culture years ago. It was William himself with a PhD student who started to develop this culture. Yeah, next slide, please. And that culture was validated in a case study in the Tessenderlo group, Tessenderlo, which is a very large, big chemical group. And on that site, we upscaled the production of the MDB of that culture, and we had very good degradation of 1,2-DCA on that site. Yeah, next slide, please. What else do we do? We do a lot of lab trials, all kinds of trials at lab scale for soil and water remediation. It says there groundwater remediation, but we do soil groundwater remediation, but we also have a lot of expertise in wastewater treatment, biogas production, anaerobic, aerobics. So we have a lot of expertise in wastewater treatment as well. So we do microcosm tests at the lab scale with biostimulation, bioaugmentation. We do a lab scale ISCO trials, so oxidation, also reduction of compounds, all at lab scale. And we, if people or industries have a certain particular problem, we design our trials really customer tailor-made for the customer. Each customer gets another kind of test. Yeah, next slide, please. Of course, this is the reason why I'm here. We have a cooperation with, we are a partner of Microbial Insights. Since 2016, we are a partner of Microbial Insights, but we, as Dora already mentioned, we used to work with Microbial Insights before that already. And yeah, we are as a partner of Microbial Insights, so we have the same quality assurance, quality standards as Microbial Insights, of course. Yeah, next slide. I mentioned it already in the beginning. We do trials, tests, and research for industries, but we also are involved in research and subsidized projects, and we work for different research projects now. But I want to highlight one of those projects. We are involved in the Electra project, and that is the flagship project of the EU-China Cooperation Initiative. And the task, I will not go into detail, but the task of Avicom in that project is that we will try to develop, we will develop a dedicated microbiome that can degrade chlorinated ethenes, TCA, DCA, and chloroethane, to ethane. So when there it's the last degradation step, which is the most difficult one. So we are also into, main thing is that we are also into research of new things. So, and this is a bit in a very, very nutshell what Avicom does. Avicom, this is a bit, I think, the most important for, that I wanted to present. Next slide, please. So we do lab trials for all kinds of industries. So if you need us, you can always contact us. And this is a bit of the partners that we were working with in the past years. Thank you.
All right, thank you very much, Lugart. That was a great presentation. All right, so we have 2 more locations, but due to the difference in time, I'm going to give you a quick introduction to these. So one of the original international locations for Microbial Insights was Microbial Insights Australia. There we have a sales representative with Hydroterra, and the laboratory that we are partnered with there is the Australian Genome Research Facility. So to give you a quick introduction to both of these, Hydroterra, as our sales representative, has the lab supplies that they can send out all throughout Australia. So anytime you have a project in that area, they have supplies on site, including the biotraps that they can ship out to you quickly for field use. Their company is really unique. It offers a variety of innovative environmental monitoring solutions such as sensors, telemetry systems, as well as data management software. So they're a great partner for us as a sales representative in the area. And then the Australian Genome Research Facility is our lab partner there. It was formerly run by one of the postdocs from Dr. White's lab, Dr. John Stevens, before he retired. And so that's kind of how that relationship came to be. Dr. Stevens was well trained in Dr. White's lab, and so it was a natural transition when he was heading up this genome research facility for us to partner with them and to develop our laboratory there in Australia. So this is some of the work that they do. They do a lot of non-profit work. They are experts in next generation sequencing, genotyping, Sanger sequencing, pretty much anything on the bioinformatics, and they're one of the world-renowned labs for this type of work. And then finally, last but not least, our location in China is with Dr. Jun Yan, who's our representative and runs our laboratory there in China. He's employed by the Chinese Academy of Science. And again, this was another relationship that was a really natural fit for us. Jun was the postdoc researcher for Dr. Frank Loeffler's lab at the University of Tennessee. So it was a natural fit for us to work with him upon his return to China. So as I close this webinar today, I just want to thank you for your attention and allowing me to really open up and share a little bit of my heart with you, to share the passion from myself, my amazing team, and our partners around the world for the work that we do here in our world. No matter where the samples come from, any industry, any geographic location, or even like those moon rocks, any other galaxy, we handle every sample with the utmost care. We know that value of the sample isn't the cost of our test, so you're never going to hear us just disregard the value of a sample. That's a fraction of the investment that you made to collect that sample and get it to us. So our staff here, as well as what you heard from our partners around the world, will ensure that your samples are handled appropriately, that the data meets the most strenuous QA/QC available, so you can always get results that you can count on. Every location around the world goes through the same rigorous training that we developed here at the laboratory at our headquarters in Knoxville. The reports will look the same from around the world, so you can easily compare and interpret data, and you're going to see something that you're accustomed to receiving. You know, 2020 has been a really difficult year for everyone around the world, and I am so proud of how our industry is continuing to care for our natural resources. We're doing this for future generations. We are so grateful for each of you and for our customers around the world, and we look forward to continuing to expand, to offer you superior customer service that no lab can match, and innovation for many years to come. So at this point, myself, the other reps that you heard from today would be happy to take any questions that you have or to answer any questions that you have about these locations. So now's the time that you can kind of really reach out around the world to see what's going on. So please enter any of your questions into the GoToWebinar software, and we will pass them out to people. Okay. So the first question that I have is about imports, and maybe I'll answer this question and then go around and let each of you talk about your shipping. And so the question is, do you face any difficulty importing samples from outside the U.S.? Does it trigger any TSCA criteria? And that's an excellent question, and it's actually the reason we founded all of these international partnerships. You know, we were receiving samples from all over the world, but we wanted to make it easy for you guys on the shipping end. So we do have all the necessary permits, soil permits, to be able to get samples into the United States. But we wanted to found each of these laboratories around the world so that you guys could easily ship the samples to whatever location is most convenient for you. So maybe I'll go around and you guys can talk a little bit about the shipping of samples and kind of what you're seeing within your country, if you're having any problems or any difficulties that you're facing with that. So maybe I'll start with you, Lutgard, since you're kind of the longest partnership in that. Are you guys seeing any difficulties within Belgium?
In fact, we collect samples throughout from all over Europe, and in fact, so far I can't recall that there has been ever a problem that we for sending samples. We collect the samples from all over Europe. We process the samples as soon as possible, the same day, filtering or extracting whatever is necessary. And then we have the protocols developed together with Microbial Insights to store samples appropriately, refrigerated, and so on. And then we send the samples, indeed, with soil permits and so on, permits required to introduce the samples to the States. And I can't recall so far that there has been any problem on that. So
wonderful.
I guess
I agree.
Yeah.
Seems like we've got it worked out, right, for the transport end.
Yeah.
All right. Okay, Stefan, how about you? Are you seeing anything in Germany that's a concern?
No, I think this works out quite beautifully with the extraction, which also will reduce the sample size considerably. So there's a speedy transport at as economic cost as possible. So it works out just wonderfully to ship eventually the extracted samples to you guys in Knoxville.
Wonderful. All right, Claudio, how about you for sample collection? You have the two labs that you're working with there in Europe. Does that seem to go well for you?
Yeah, we had no problems so far. I mean, our clients ship the samples directly to Abercorn in Belgium. It takes normally 1 day to get there. So they just have to collect and store the samples correctly and ship them to Abercorn. So, and normally it takes 1 day. So no problem about that. Yeah.
Great. All right, and last but not least, Will, what are you seeing in Canada as far as shipping?
No issues from an MI standpoint, as long as all the customer documentation's correct, which we included. It's overnight delivery. We typically ship it out to parts EBPI around 4 o'clock, 4:30, and I usually notice it arrives around 9:30, 10:30 down at MI the next day. So it's less than 4 hours.
Perfect. All right. And I think if you guys have questions on that end, feel free to reach out to Jennifer Amos in our customer service department. There's no one that has a better relationship with FedEx and being able to get samples sent all over the world and knowing the paperwork than her. So if you have questions, definitely reach out to her. One last question for each of you before we wrap up the webinar today. Can you talk a little bit about the state of what's going on in your area with remediation projects and kind of what you're seeing? I know 2020 has been a difficult year and a lot of change for all of us. And I'm curious and would love to hear what you're seeing in your location. So if you can we'll end the webinar today kind of talking about each country, if you don't mind. So Will, do you want to go first? Talk about Canada?
Sure. What I'm seeing right now is we're sort of still in a bit of a lockdown in Ontario. Canada, we're allowed to hang out with 10 people now, which is good. Travel's starting to open. The issue right now with the lockdown is to obtain the field samples. You're going to have to travel. Canada's the second largest area size country in the world. So there's a lot of distance to travel to obtain samples. So that's really, until we start to see a bit more travel, getting people out there, we'll start to see that pick up shortly, I would think, as we open the country.
All right, thank you, Will. So Claudio, can you talk a little bit about Italy and what you're seeing now as industry starting to reopen?
Yeah, the environmental market in Italy is quite sparkling at the moment. I mean, Italy is the first first country in Europe with the biggest turnover in environmental market, driven mainly by private sector. So not many public funds, but mainly private funds. And we are in these years we see that we move to in situ treatments instead of pump and treat and dig and dump. So we are moving to in situ and more green treatments. So it will be also good for biological treatments at the end. I think we need more professionals in this field in Italy than before, because the treatments will be more complex. But yeah, we can do that. We need also the support of the foreign companies like you, because you have more experience, you made more experience in the past years than Italian companies. So you can find in Italy a company like yours. So that's why I looked abroad. Yeah, and also other companies like Provectus, Ivy, that are our partners are for that. Yeah, that's it.
All right, thank you, Claudio. Stefan, what are you seeing in Germany?
Yeah, fortunately, there has never really been quite a strict lockdown that would, prevent any industrial activity. So we have been lucky to, or fortunate to, be able to move around in the country to follow up on the remediation projects. I think most of these projects have been planned and worked for on a long-term basis. So it's really hard to stop these activities. In fact, the authorities made sure that remedial activities would still be worked on, and we're actually seeing now a lot of business opportunities with all the funding opportunities that are being released to help the industry. So we think it's actually an industrial sector. The remediation business is a sector that has not been hit by COVID-19 as hard as other businesses have.
All right, thank you, Stefan. And then finally, Lugart, can you talk about what you're seeing in Belgium with the restrictions and what's happening there?
Yeah, so in Belgium, COVID-19 was quite severe as well after Italy and Spain. I think Belgium was the third country in Europe. And in fact, so there has been a lockdown for 1.5 months. And since at the start of, since beginning of May, gradually Belgium started its exit strategy. So gradually we were allowed to do more, and we could go out gradually more and more. Companies could open again mid of May. And well, I must admit that I estimated that this COVID-19 would have had enormous impact on the economy, and that will be, of course. But I see now that the impact of COVID-19 on, for example, soil remediation will be a rather small impact. I mean, we already now, I told you that we are also doing a microcosm tests, lab scale trials for soil remediation. And beginning of June, we received again two confirmation that 2 tests will go on. So when we will start next week, we will start a new microcosm test. So this means that that companies can go on site to get groundwater samples and to get soil samples. So in fact, it's my impression, I feared that would be a serious impact, but I have the impression that soil remediation, all those things will recover quite soon. And I have the impression that maybe we are not working already at full speed, but it's nearly 100% recovery, and I mean in activity, not in budget, of course, but in activity, I think we are we are again quite normal. So yeah, this looks good. We didn't expect to get orders for microcosm tests so fast after the lockdown, but we had 2 of them, so it looks like yeah, soil remediation, it restarts, in fact.
That's wonderful. Well, first of all, thank you all as affiliates for being here today and being able to share with us kind of the state of your country, what you're doing, and the state of your companies. And I appreciate all the work that you do with us, and I look forward to many years of partnership and working together in this industry. So I'm grateful for you for being here and taking time. I know that the time change is not always the best when we're looking at the U.S. versus Europe. So I appreciate that you guys took time to come and be on the webinar with us today. And for the audience, we are very grateful for you letting us share a little bit of who we are and where we're going. And if you have questions or if you need help on projects, reach out to me or to anyone on my team. Or to any of the affiliates in different countries. We are honored to work with you guys and to help you and look forward to serving you for many years to come. So we're going to be doing a recording of this webinar. It'll be available on the resource section of the website. I've had a few of you email and ask about that. Also, if you want to email info@microbe.com if you have additional questions, or if you would like a copy of the slides, we'd be happy to send those out to you. But again, we appreciate your time, and we look forward to having you join us on our next webinar. And thank you, everyone, for joining and doing the webinar with me today. I really appreciate each of you.
Thank you, Dora.
All right, thank you. Thanks, Will.
Thank you, Dora.
Thanks, Claudio. Thanks, Stefan. Thank you, Lucart.
Thank you, Dora, for the opportunity to present ourselves. It's been very nice.
All right, thank you. You guys have a wonderful evening.
Bye-bye.